Hello, my name is Mohammad Rashad, and today I'll be presenting about inhibiting the effects of chronic lymphocytic leukemia using ibrutinib and mithrimycin. I worked under the mentorship of Dr. Subro Bandada at the University of Kentucky in the Department of Microbiology and Immunology. So first, to give a general overview of what I'll be presenting about, first I'll give an introduction and background of my project. I'll then present my question and hypothesis. I'll then discuss the methodology and procedures behind my experiments. I'll then discuss my results and what they mean for my project overall. And I'll then discuss and conclude my project. And I'll then talk about future research and the next steps for my project. And I'll end with some acknowledgements for people who have helped me over the past 2 years and present my sources. So first, to give a general introduction, my project revolves around chronic lymphocytic leukemia, or CLL, which is the most common type of leukemia in the Western world. And this type of leukemia starts in B cells called lymphocytes, hence the name chronic lymphocytic leukemia. And these cells require constant stimulation through a B cell receptor in order to survive and to proliferate. These cells usually start in the bone marrow, but then eventually move their way into the bloodstream and spread throughout the rest of the body. And as you can see in figure 1, this is a representation of what these cells look like as they start to accumulate in the bone marrow. And in figure 2, this is what they look like when they start moving their way into the bloodstream and spread throughout the rest of the body. Now, the current treatment for chronic lymphocytic leukemia is a drug named ibrutinib. Now, ibrutinib targets a signaling protein downstream of the B cell receptor called BTK that allows these cancerous cells to survive and to proliferate. So by targeting BTK, this in turn halts the survival and the proliferation of these cancerous cells. Ibrutinib also reduces the ability of CLL to secrete a factor called IL-10, which is a cytokine that doesn't allow the immune system to fight off against the cancer. So by inhibiting IL-10 production, this in turn actually allows the immune system to combat chronic lymphocytic leukemia. So this call kind of shows the positive effects of ibrutinib, but the main issue with this drug is that many patients eventually acquire resistance to it after a couple months of treatment, so it isn't a very effective long-term treatment for chronic lymphocytic leukemic patients. And as you can see in figure 3, this kind of shows the function of ibrutinib before and after patients begin to develop resistance to this drug. So before resistance, you can see that ibrutinib targets BTK, which in turn halts the proliferation and survival of these cancerous cells, while also reducing IL-10 production, which, as I mentioned earlier, allows the immune system to actually fight off against the cancer. However, when these patients begin to develop resistance to ibrutinib, there's a mutation in BTK that doesn't allow ibrutinib to function correctly. So as a result, these cells continue to survive and to proliferate, and IL-10 is still alive and present, which unfortunately is how many patients end up dying because the immune system isn't able to fight off against chronic lymphocytic leukemia. So it's already known that a drug named mithrimycin inhibits IL-10 production by targeting the transcription factor called SP1 that's responsible for IL-10 production in the immune system. So by targeting the initial source of IL-10 in the immune system, mithrimycin is able to inhibit IL-10 production and actually allow the immune system to fight off against CLL. However, mithrimycin has not been tested with CLL or as a potential alternative treatment for chronic lymphocytic leukemic patients. And as you can see in figure 4, this kind of shows the function of mithrimycin. As you can see, it completely bypasses BTK and instead targets SP1, which, as I mentioned before, is the transcription factor responsible for IL-10 production. So by targeting SP1, this in turn reduces the production of IL-10 and allows these cancerous cells to stop the proliferation and survival while also letting the immune system fight off against the cancer. So essentially my question and my hypothesis, my initial question was what effect will mithrimycin and ibrutinib have on human CLL cell viability and IL-10 production? My initial hypothesis was that mithrimycin would be able to reduce the viability of CLL cells while also reducing IL-10 production. I'm moving on to my procedures and the methodology behind my experiments. The first experiment that I ran was a cell culture using ibrutinib, mithrimycin, and a combination of both ibrutinib and mithrimycin at varying concentrations on a normal human CLL cell line. The next experiment that I ran was a residuant viability assay in order to determine the number of living and working CLL cells. And the way that a residuant viability assay works is that you insert a blue dye and residuant into the mitochondria of these cells. So you essentially insert a blue dye into all these cells, and after a certain incubation period, the dead cells and the living cells turn into different colors. So by measuring the fluorescence of all these cells after this incubation period, I was able to determine which cells are still alive and which cells are dead, and what effect mithrimycin, ibrutinib, and a combination had on all of these cells. So this is how essentially I determined CLL cell viability. The next experiment that I ran was an ELISA lab, or an enzyme-linked immunosorbent assay. And essentially, ELISAs are very commonly used to measure cytokine production. And for this experiment, the cytokine that I was interested in was IL-10, because as mentioned earlier, IL-10 doesn't allow the immune system to fight off against the cancer. So by measuring IL-10 production, this in turn allowed me to see what effect mithrimycin, ibrutinib, and a combination had on reducing IL-10 production. And as you can see in figure 5, this is an example of what the cell plate looked like during the ELISA experiment. And figure 6 is a picture of a flow cytometer, which is the machine that I used to actually measure IL-10 production during ELISA. So moving on to the actual results of my experiment, I ran 5 different trials for the viability assay. One was unsimulated, and then I used 4 different simulants just to see if the simulants would have any positive effect on reducing the viability of CLL cells. And as you can see in figure 7 was the unsimulated trial. And ibrutinib was the blue line on this graph, mithrimycin was the orange line, and the combination was the gray line on this graph. And then I used 2 different simulants, DMSO and TRINE-X, or TX. And TX is actually a liquid detergent that kills off all of these cells. So that was my negative control for this experiment. And DMSO was my vehicle control, so that's essentially what I compared everything to during my experiment. As you can see in figure 7, mithrimycin and ibrutinib, mithrimycin and combination were actually more effective than ibrutinib in reducing the viability of these cancerous cells. And you can see in figure 8, when treated with a stimulant called LPS, or lipopolysaccharide, mithrimycin and a combination were once again more effective than ibrutinib in reducing the viability of these cancerous cells. And in fact, you can see that in figure 8, mithrimycin and a combination were so effective that the effects of both of these treatments were equivalent to the effect of when these cells were treated with TRINE-X or TX. And as I mentioned earlier, TRINE-X actually kills off all these cells. So this tells me that in this specific trial, mithrimycin and a combination were so effective that they actually ended up killing off all these cells. So that's actually a positive thing for my project overall. Moving on to the rest of the results for the viability assay. In figure 9, I used a stimulant called poly-IC. Figure 10, I used CPG, and figure 11, I used a stimulant called anti-IgM. And in all 3 of these trials, as you can see, mithrimycin and a combination were once again more effective than ibrutinib in reducing the viability of chronic lymphocytic leukemic cells. And once again, you can see that in figure 10, when using CPG as a stimulant, mithrimycin actually overlapped TRINE-X at a specific concentration, which tells me that mithrimycin was so effective in this specific trial that it actually ended up killing off all the cancerous cells at a specific concentration. So again, this is very promising for my project overall. As for the ELISA experiment, the results of this experiment showed that mithrimycin, ibrutinib, and a combination were all effective in reducing IL-10 production. Mithrimycin was the red line on this graph, ibrutinib was the blue line, and a combination of both ibrutinib and mithrimycin was the green line on this graph. And the control that I used for this experiment were untreated CLL cells, so cells that were treated with neither mithrimycin nor ibrutinib, and this was the purple line on this graph. However, there really wasn't any distinction between mithrimycin combination and ibrutinib, so I wasn't able to really say if there was any difference between mithrimycin, ibrutinib, and a combination or any of these treatments in reducing IL-10 production, but I do know that all 3 were effective in reducing the production of IL-10. So what do these results actually mean for my project? First, my hypothesis was proven to be true and accepted that mithrimycin was effective in reducing the viability of CLL cells while also reducing IL-10 production. However, there could have been some possible errors with my project overall. And one possible error is simply human error, which could have led to variability in the assay results that you see in these graphs. Another possible error could have been a contamination problem in my lab last year that could have had a minor effect on my results as well. However, the major conclusions of my project was that mithrimycin was effective in reducing the viability of these cancerous cells while also reducing IL-10 production. And this overall shows a lot of promise for mithrimycin as a potential alternative treatment for chronic lymphocytic leukemic patients in the future as they begin to develop resistance to ibrutinib. So for further research, one of the next steps for my project is to see what effect mithrimycin has on the rest of the immune system. because as I mentioned before, chronic lymphocytic leukemia mainly deals with B cells, cancerous B cells in the immune system. However, there are obviously many other factors in the immune system that contribute to the productivity of the immune system overall. For example, T cells. So seeing what effect mithramycin would have on T cells would give me a better indication of whether there are any negative side effects of mithramycin on the immune system. Another next step for my project is seeing what effect mithramycin would have on ibrutinib-resistant CLL cells instead of just a normal human CLL cell line. Because as I mentioned before, for this experiment, I used to use a normal human CLL cell line, but my main original question and my main original goal for my experiment was to see what effect will mithramycin have on CLL cells as they begin to develop resistance to ibrutinib. So to actually do this, I will first use polymerase chain reaction, or PCR, to replicate and amplify the genome. I'll then use bacterial transformation to actually introduce a mutation in BTK, which induces resistance to ibrutinib. So this will actually in turn give me ibrutinib resistance to CLL cells, and then I'll repeat these experiments with this ibrutinib resistant cell line, and this will give me a better answer and a clear answer to my goal and original experimental question. And another, finally, next step for my project that I can do in the future is actually seeing what effects mithramycin and ibrutinib in a combination would have on IL-10 production with different stimulants. Because as you probably noticed on my ELISA results slide, I only used one trial for this experiment with no stimulants at all, using ibrutinib mithramycin in a combination. However, using different stimulants could give me a better indication of what effect these stimulants could have on IL-10 production, and if they could increase the productivity of these treatments on IL-10 production. So that's essentially it for my project, but I'd like to acknowledge and thank some people who have helped me over the past 2 years. First, Dr. Subur Bandada, who actually allowed me to work in his lab over the past 2 years and mentored me. Dr. Jacqueline Rivas, who's a postdoctoral candidate at Dr. Subur Bandada's lab, who guided me and helped me over the past 2 years. Dr. Sarah Hakim, who's also a postdoctor at Dr. Bandada's lab, who also helped me a lot over the past 2 years with my project. I'd like to thank the University of Kentucky and the MCC program for granting me the opportunity to actually work in Dr. Bandetta's lab, and the same goes for the Kentucky Young Researchers Program. Finally, I'd just like to thank family and friends for giving me rides to and from lab over the past 2 years and just encouraging me with this project overall. Here are my sources, and if you have any questions, please let me know. Thank you.